X-linked retinitis pigmentosa is a severe inherited retinal disease driven by mutations in the RPGR gene. It follows a relentless clinical course, starting with childhood night blindness and progressing into a severe constriction of the visual field. The X-Alaris Natural History Study tracked this progression over 24 months, documenting the persistent thinning of the outer nuclear layer. As these photoreceptor cells die off, the patient's field of vision steadily collapses. This cellular loss has traditionally defined the limits of ophthalmic intervention. The foundational assumption was that gene therapy could only serve as a stabilizer, a way to halt further cell death. Since dead retinal tissue was considered impossible to rebuild, the clinical goal was restricted to freezing the disease in its current state through early intervention. This assumption was tested in a first-in-human phase 1-2 trial using an AAV8 viral vector to deliver a corrected version of the RPGR gene. The trial produced a result that challenged the stabilization model, evidence that at optimal doses, the therapy could drive a measurable increase in the thickness of the retinal architecture. Securing this structural recovery requires navigating a narrow therapeutic window, which is strictly bounded by the risk of subretinal inflammation. The success of the treatment hinges on a precise calculation, finding a dose high enough to rescue dying cells, but low enough to avoid a viral load that triggers immune toxicity. The native RPGR gene is notoriously difficult to work with. It contains a repetitive, purine-rich region that makes the sequence unstable, often leading to alternative splicing errors during protein production. To bypass this instability, researchers codon-optimized the sequence. This stable core RPGR variant maintains its fidelity and is then packaged into an AAV8 viral vector for delivery. The vector is delivered directly to the target cells through a two-step subretinal injection. This process creates a localized fluid bleb beneath the retina, covering the posterior pole and temporarily detaching the fovea to ensure maximum coverage of the macula. Because this delivery method is geographically isolated to the bleb area, it allowed researchers to run a highly controlled dose escalation experiment within the eye. The trial began with low vector concentrations in cohorts 1 and 2, delivering up to 1 × 10 to the 11th genomic particles per milliliter. While these low doses were safe, they produced no measurable functional improvement in the patient's vision. These early participants had very advanced retinal degeneration at the start of the study. When the vector dose is low and the cellular infrastructure is already highly compromised, the remaining cells lack the metabolic capacity to restart their function. The results shifted significantly with cohort 3, which received a mid-range dose of 5 × 10 to the 11th genomic particles per milliliter. These microperimetry heat maps track retinal sensitivity. By month 1, all 3 patients in this cohort showed an immediate expansion of their visual field. This improvement was sustained through the 6-month follow-up, with the high sensitivity zones remaining stable. While the functional gains were clear, the researchers looked to objective structural imaging to see if they could find the biological cause. Manual segmentation of the OCT scans confirmed the disruptive finding. The thickness of the outer nuclear layer had actually increased. The scans also revealed the reemergence of a new linear anatomical structure at the distal photoreceptor level, the area where light is physically converted into signals. This structural reemergence is evidence of active biological restoration. It demonstrates that with the correct viral load, the retinal architecture can physically recover from a state of advanced thinning. As the trial pushed into higher concentrations, up to 5 × 10 to the 12th particles, a new complication appeared. Patient C4.1 initially saw excellent functional gains. However, at week 5, his retinal sensitivity suddenly collapsed back to 0. The OCT scans at week 5 revealed scattered subretinal lesions, appearing as hyperreflective clumps exactly where the vector had been deposited. These lesions were a localized immune response, a steroid-responsive inflammation, triggered by the high viral capsid load in the subretinal space. Treatment with oral prednisone cleared the inflammation. By week 12, the patient's sensitivity rebounded fully, eventually reaching a peak of 6.6 decibels. While this specific patient recovered, the same inflammatory response permanently offset visual gains in other high-dose participants, defining the absolute toxicity limit of the therapy. These results show that the success of RPGR gene therapy is a balance between the stage of the patient's degeneration, the vector dose, and the immune response. We can visualize this balance by plotting the trial data into a therapeutic index. On the left, we have the low-dose cohorts. The vector concentration is safe, but it's insufficient to drive cellular rescue. On the right, the efficacy drops off sharply as we enter the danger zone, where the high viral load triggers destructive inflammation. The peak represents the mid-dose used in cohort 3, the Goldilocks zone, where the vector concentration is high enough to stimulate recovery, but low enough to avoid the immune threshold. Identifying this precise therapeutic window proves that X-linked RP is not just a disease that can be halted, but one that can be structurally reversed. This dosing architecture provides the foundation for the final curative trials.